Genetic code consists of 64 codons, which altogether code for 20 amino acids. You're not expected to know all of them, but some key ones to know are your start codon, which are AUG or ATG. This is also called methionine. And then you've got your stop codons, which are either your UUA or TAA, UAG or TAG, and then UGA or TGA. All amino acids have the same basic structure, so you've got amino, which is your N-terminus, you've got a carboxyl group, which is your C-terminus, you've got hydrogen, and then you've got an R or variable group. It's this variable group that gives each different amino acid its unique chemical properties, and that's why we have 20 different ones. So we use the genetic code that's brought across by the mRNA to put these amino acids in order. So you've got 3 steps in this translation of the mRNA. Your first one being initiation, which is when the ribosome assembles around the mRNA and the tRNA attaches to the start codon. Your tRNA has a complementary anticodon to the mRNA, and I'll go over the structure of the tRNA in a minute. Your next stage is elongation. This is when the tRNA transfers the amino acid that it's brought, and then the next corresponding complementary tRNA binds as well. Then you have translocation, which is when the ribosome moves along the mRNA chain so that more codons are available for the anticodon of the next tRNA to join. This causes an increase in your amino acid chain as peptide bonds are forming between them as they go along. Your final stage is termination. This is when the ribosome actually reaches the stop codon and causes the newly synthesized amino acid chain to dissociate due to a release factor. So the tRNA looks a little bit weird. It's sort of all bent and there's bonds forming between each of the bases, even though it's just one linear RNA chain. And then there's actually an amino acid that is specific to its anticodon joined at the opposite end. This is joined by an ester bond. I did mention ribosomes when I was going on about the organelles in prokaryotes and eukaryotes videos, but we'll just give a little brief overview here. It's often called in my class the hamburger because it's made up of a large subunit and a small subunit, which is made by proteins and rRNA, but your large subunit in eukaryotes is 60S, whilst your small subunit is 40S. So there are 3 different sites within a ribosome. You've got your A site, which holds the amino acyl tRNA, so it's tRNA with the amino acid attached to it. You've then got your P site, which holds the tRNA that attaches to the growing polypeptide chain, so the amino acid is sort of forming a bond with the previous amino acids. And then you've got your E site that's holding this tRNA that's already lost its amino acid and then will be removed.